Yeah, thanks for hanging out so long, and thanks for coming to the conference. It was a real blast. I loved it. And now you have to hang in with me for a while, and I hope I can entertain you a bit. The bus is coming, hopefully.
Time until the bus is here.
I don't make predictions on that. So sorry for that. So I picked a title which is a bit broad. It says Designing Functions in Synthetic Cells. And what I really mean is that there is place for theory in the research of synthetic cell. And I'll try to explain why I think so. Let me start with a few things where you just should be in awe how beautiful and complex biology is. So this here is cell polarity in yeast, where you have the formation of a polar pattern of proteins, and that polar pattern of proteins then recruits actin filaments, and these actin filaments then create the bud site. So the function, the clear function of that protein pattern is budding, so cell division in the end. Same is true in E. coli. In E. coli, you have pole-to-pole oscillations of proteins called min proteins. They have a similar function. They define midplane so that the cell knows where to divide actually 50-50 into two daughter cells. Then here's work I did together with Nikta. That's the starfish oocyte from a different perspective, from an inside perspective, where you see pattern formation. And this pattern formation there is in a very, very nice way linked to the morphology of the cell. So you have a coupling between differential geometry, if you like, and nonlinear dynamics. And last but not least, here is an experiment by Petra Schwille's group that shows really just a vesicle, not a cell, right? This is just a hollow shell of a lipid bilayer on top of a protein pattern, and this thing moves. And I would say this is active, right? But the reason for it being active is not that you have self-propulsion by any means. It's really the transduction of chemical energy into patterns, and those patterns then are able to, through some mechanism, create mechanical forces that move the vesicle. Now, this is the kind of long-term vision. We have seen these beautiful phenomena in biology. Question is, can we, at least in a very rudimentary way, reconstruct these kind of features in a synthetic cell? And I would say, yes, we can actually do this, and an important role will be played by theory. And the reason I think it will be played by theory is because the parameter space and the topology space of chemical reaction networks is so huge that it just doesn't really make sense to try and make this all on the basis of trial and error type of construction and design. So here I think is the minimal system where this could really work. The minimal system, I think, is just a vesicle, so a lipid bilayer, and in this lipid bilayer, you do have some proteins. And I think this works because it combines mechanics and geometry. So you have membranes and proteins. There's a mutual feedback between the two. You can have the pattern reading off the geometry of the vesicle. So it encodes a spatial structure depending on the vesicle geometry. And then you need, as a second part, that the spatial structure encoded by the protein pattern is actually able to change the geometry. So if you take things together, there is a list of things that you need in order for this minimal system to actually work in some way as a synthetic cell, meaning it's dividing or moving or doing something of that nature. The first thing I think we need is a molecular and conceptual understanding of protein patterns. It's not enough to have a mathematical model of the pattern formation. That wouldn't help you. You need the connection from the pattern all the way to the molecules and their molecular properties, because in the end, you would like to make design suggestions to experimentalists that then are able to design the proteins in a proper way such that they perform in a way that creates a certain protein pattern. And also conceptual, but I'll talk about this later. I said this, the protein patterns need to be able to sense and respond to the geometry. That implies that the pattern cannot just be localized on the membrane boundary. The pattern has to penetrate into the bulk of that protein, of that vesicle, because otherwise it wouldn't be able to sense the external geometry it's embedded in. It needs to change the geometry, I said this, because otherwise you don't have the force element that you need in order to have shape transformation. And you can also say there's some kind of mechanochemical coupling that creates forces. This doesn't need to be only the ability to create spontaneous Gaussian or mean curvature. When you saw the vesicle moving on a substrate, you also need the ability of these patterns to somehow create adhesion forces between the bilayer on the substrate and the vesicle that you have on top. All right, so that's what we need. And now let me go through these different points step by step, and let's see how far I get in these 25 minutes. Molecular understanding of patterns, I think we are there. So these are E. coli oscillations. Nowadays, you can measure those E. coli dynamics in vivo, in vitro, in all kinds of reconstituted systems. There's a whole armada of measurements that you have on these kind of systems. And recently with Suk-Yoon Yoon, he managed to actually manipulate the E. coli cells such that he could get a phase diagram, so change the protein concentration to see how the patterns change in the E. coli cell. So let me recap what these patterns mean to do. They mean to define midplane, the division site, by these oscillations that go back and forth. And if you average over the oscillation, you get some profile that has a dip in the middle. The reaction circuit that drives that was supposed to be this. I think Ned Wingreen and others came up with that early on. And the idea is basically like that. Here, this is the unit where you drive the system off equilibrium. It's an ATPase, where you change an energy poor form in an energy rich form, and only the ATP-bound state can bind to the membrane. And then you start saying there is recruitment, meaning positive feedback, that drives more of those proteins to the membrane. And then there's a min-E protein, a second protein, that then is also recruited to the membrane. They form a dimer. And once they form a dimer, the min-E is hydrolyzing the min-D, making it deactivated again. So in that way, you create a reaction cycle. And by having that cycle, and Kolmogorov's ideas, right, this is a broken detailed balance system driven by non-equilibrium fluxes between the membrane and the cytosol, and also between different kind of states. But is that the right model? Is this really keeping the promise that we do have a molecular understanding of these pattern-forming systems? That's the first thing I would like to go through with you. This is the machinery. I will not talk much about how we actually do the theory, but just to be clear, this is diffusion reaction dynamics. There's diffusion reaction equations in the bulk. There's diffusion reaction equations on the membrane. And there's boundary conditions. They are such that any kind of gradients into the cytosol has to be fed by reactions that come from the membrane or go towards the membrane, such that the overall system that you have here conserves the total mass of proteins that you have. Right now, let's check this theory, where I said we have min-E and min-D giving rise to protein patterns. And I didn't tell you, but one key prediction of that theory was that in order to get patterns, you need to have less min E proteins than min D. Right? Because otherwise, if you have too many of those min E, which are antagonists, the whole pattern wouldn't happen since it suppresses the formation of min D going towards the membrane. And when you look here into these data from Petrashvili's group, there's obviously a contradiction, right? Theory says the concentration should be less than 1. It's actually forming patterns over a very, very broad range of parameters. So that means the theory is flat out wrong. It doesn't explain the data on a molecular basis. But one could rescue it, one can rescue it by taking into account, which sometimes in statistical physics you might consider as a minor detail. And this minor detail is that the min E itself, this protein, is a switch. It goes up, it opens and closes on a microsecond time scale. The pattern is on a minute time scale. Right? This is really details. But the point is, this is a diffusion reaction system, not in bulk. It's a diffusion reaction system that's coupling the bulk to the boundary. So the real thing is not a time scale. The real thing that matters is the length scale, is the vicinity, the layer thickness of the boundary at the membrane. So you have to take into account this min E switch. And then to make a long story short, this min E switch does the job. The theory tells you that this acts like a latent state, like a kind of reservoir, which you can tap into, and that makes the system more robust to changes in the relative ratio of min E to min D concentrations. So if that were to be true, you could tell the experimentalist, well, just switch off the switch, right? And then you should see that the pattern loses its robustness. And that's what Simon Kretschmer in Petra's group actually did. And indeed, when you inhibit the switching in the min E, it shrinks down to a very narrow window, and you lose all the robustness that you have in pattern formation. This is an in vitro experiment, so a reconstituted experiment where you have in control all the concentrations, and a biologist wouldn't really believe you if you show these kind of data. They would ask for in vivo experiments, and that's what Suk-Yun Yoon did over the last basically 4 or 5 years. So that was quite an endeavor from his group. And what they found, and I guide you through quickly, is a phase diagram where they were able to vary the concentration of one protein, the min E, but also the concentration of the other protein, min D, over was something like 2 orders of magnitude, roughly. And they find that there's a large regime where there's no patterns, no oscillations. And then there's this broad regime here in bluish and greenish where they find pattern formation, different kinds of patterns. They find traveling waves and standing waves. And because of regulating the biological systems, some of these cells grow large, some are small. And that's what you see here. You have longish cells, you have shorter cells in different regions, and they have all kinds of different oscillations. So the point was, can we recapitulate that with our model in quantitative detail? And for that, they needed to use a drug, and that drug was responsible for making all the E. coli cells equal length. Okay? Once you have that, you can actually compare theory and experiment on a quantitative level. So here is the experiment, the theory, the simulations, and that's here is the experiment. You see it shows the same kind of phase diagram. It's on an absolute scale, so we're not fiddling parameters. This is matching it on an absolute scale. And then you have the green region and the reddish region here and in the simulation as well. And that distinguishes standing wave patterns from traveling wave patterns. So there's a nice correspondence. And one slide that I skip is that you can actually compare the period and the wavelength. And that also matches on a quantitative basis. So I would say from that, we do have a molecular model that explains the data quite well. And now let me go and step back a little bit and look at the network from a conceptual point of view, because I think these networks are modular, and different elements of those networks have very specific biological function. There is the basis network, or skeleton model as we call it. That's the one I explained at the very beginning, and it does the job. I would consider this a kind of mathematical model that reproduces all the phenomena that you can see. It's kind of a universal type of model that reproduces spiral waves that's seen in vitro experiments, traveling waves, standing waves, all of that. But it lacks two key features. It lacks robustness, and it lacks to have the quantitative agreement. It misses the length scale by a factor of 10 or 20 or something like that at least. So for that, you need other modules, and I just explained to you that the switching of the min E protein opening and closing is the module in that network which conveys a robustness to the pattern-forming system such that the biological system really works fine over a broad parameter regime. There's another module which is already indicated here on the graph. This is what we call persistent min E binding. So it's indicated here. Once you have the hydrolysis where the min E is inactivating the min D, they could fall off both from the membrane right away, right? They could just go back to the cytosolic state, but there is one type of network where they could actually stay on the membrane, and that's the key feature that you need in order for the system to get a robust length scale that really agrees with the experimental data. And once you have these things together, then you actually have this molecular model that we wanted. There's one little question that remains, namely when I say recruitment, I guess none of you knows what recruitment means, or everybody has some idea of what it could mean. What it means in chemistry is there's feedback. There's some nonlinear rates that you have to include into the biochemical model. And recently, and it's not published work, we figured out that the reason for recruitment is actually a phase separation phenomenon, not phase separation in the sense that you have molecules that glue to each other in an isotropic way, more like sticker molecules, and these sticker molecules arrange themselves in a some kind of longish polymer or star-like shape, and that's the physical mechanism that gives rise to the nonlinear feedback. Now, conceptual understanding of patterns. You can see pattern formation as a black box. Just say you have diffusion reaction equations, you switch on your COMSOL simulation, and you get a pattern, and it agrees with data or it doesn't. You can also ask, when you look at these pattern-forming systems, are there new phenomena that you could exploit because they give rise to new kind of supramolecular patterns that you could exploit in structuring the features that you have on a synthetic cell? So for that, the questions we ask is whether these pattern-forming systems could actually have some structures that you usually find just in mixtures or in foams. And for that, you want to ask whether there is some mechanism that underlies the formation of these structures. And when you know about equilibrium foams and equilibrium mixtures, then you know that the key ingredient for all of those structures is that you need a surface tension. Okay? So is there something like a mesoscopic law that gives rise to surface tension in patterns? That's a weird question, because a diffusion reaction system is a gas-like system. It has a low density of particles. There is no no pairwise interaction in the sense of molecules attracting or repelling each other. It's only chemical reactions that take place. So there must be a different reason that explains surface tension if it exists. So here is data from C. elegans zygotes, and this is simulations from Rafaela Gesele in my group some time ago. And what you should notice here in the simulation is that the pattern forms and then realigns, and it realigns in such a way that the interface length that you see there is going to a minimum. And that's a clear feature of a system that shows line tension, because otherwise you wouldn't have something that tries to minimize the interface length. So where does that line tension come from? Well, in equilibrium physics, there is a clear and well-known answer from just thermodynamics. Thermodynamics tells you there's the Gibbs-Thompson relation. This Gibbs-Thompson relation says there must be a chemical potential difference if there's a surface tension and a curvature, given that you have a jump between a high density and a low density phase. Physics of that is just the fact that as you curve an interface, you have less interactions depending, or less or more interactions depending on the curvature of that interface. And that says then there is a chemical potential difference given on that curvature. The chemical potential difference creates a current. This current is such that the initial perturbation is attenuated, and you go back to a flat interface, which is nothing but surface tension, making things flat or decreasing the length of interfaces. So why should there be something like that in a non-equilibrium system? And here is my pictorial way of explaining that. Behind that, there's a singular perturbation theory that you can perform, matching inner and outer regions of these patterns. But let me just wave my hands and explain how that works. So I told you the key feature of all these protein diffusion reaction systems is that you have a flux from the membrane to the cytosol and back between different states. Okay? And this flux is what maintains interfaces between high and low density regions on the membrane, such as here where you have a high membrane density going to a low one, and the corresponding density in the cytosol going from low to high. And as said, the interface itself is maintained by a non-equilibrium flux driven by the ATPase Mindy reaction cycle. There is an attachment flux on one side of the interface. There's a detachment flux from the membrane back to the cytosol on the other side of the interface, and counteracting these two fluxes are diffusive fluxes in opposite direction here on the membrane and there in the cytosol. Okay, now you have this interface zone with attachment and detachment zones, and now imagine looking on top and bending the interface. And then there's a very simple geometric argument. As you bend an interface, which has a certain width, you will stretch the outer part of the interface and you will compress the inner part of the interface, depending on what kind of curvature you have. So this curvature here will stretch the outer zone and compress the inner zone. If you have the curvature going the other direction, you just do the opposite. You compress the detachment zone, but you extend the attachment zone. All right, once you have that, it's pretty clear, because the fluxes will be proportional to that area, that here in this region you have more detachment, and here in that region you have corresponding more attachment that happens to the membrane. And therefore there will be a surplus or a lack of proteins in the corresponding cytosol. You have more proteins here, you have less there. You have a gradient in concentration. Gradients in concentration give rise to fluxes. Those fluxes are directed such that you fill up the gap here. So again, you straighten out the interface. The same kind of argument as before. It's just not the pairwise interaction between particles being responsible for that kind of gradient. It's the imbalance of non-equilibrium fluxes at regions in your system that do have different curvature. So if you put this into equations, this gradient here is the change in area due to curvature related to the total area, and by simple high school geometry, this is the width of the interface times the curvature that you have that bends the interface. And that's the same law as the Gibbs-Thompson relation that I showed you. It has the same structure, and it means that the interface tension created by non-equilibrium fluxes are related to the interface width. The interface width is determined by diffusion coefficients, because diffusion coefficients together with reaction rates define a diffusion length, and that's the length scale that you have here that gives rise to the interface tension. So in a nutshell, non-equilibrium fluxes give rise to non-equilibrium surface tension that act in the very same way as surface tension that you might see in a thermodynamic system. Because you have surface tension, you should be able to get all the phenomena in equilibrium statistical physics that are based on surface tension. So you should see physics of mixtures, you should see the physics of foams, you should see all the laws of mixtures and foams that are just based on the fact that you have surface tension. And indeed you do, and I skipped the part on mixtures, and let me directly go to the part on foams. So here you see data again. Those data are from Petra's group. They have made those measurements in 2019, so quite some time ago. And they didn't really have any kind of explanation on these patterns at that time. And we had neither. We just ignored these kind of patterns. But after we realized that there is surface tension in these diffusion reaction systems, we were like, oh, this must be foams. Okay? If these are foams, then they should obey the foam laws. One famous foam law, of course, are the plateau vertex conditions, meaning that you have to have 120-degree angles at all the vertices. And indeed, you see, you get 120-degree angles with a very small standard deviations at these protein patterns. Another foam law is that foams coarsen. Pity for all of us who drink beer because foam goes away, right? Here it doesn't. This type of foam is not coarsening. It has an equilibrium distribution of these cells that is a stationary distribution. So in addition to coarsening of the foam, there's a counteracting process that is anti-coarsening of the foam. And there must be a reason of why there is anti-coarsening in these kind of foams. So I told you that we do have a quantitative molecular model of the min patterns. And that means we can also use it to actually mimic or even reproduce, because it's molecular, the phenomena that's seen in an experiment and look at them in more detail, in a detail which you couldn't do in experimental analysis. So what Hendrik did is to take an equilibrium structure of such a foam on a computer and scale it down such that you are off the fully coarsened state, so the system must coarsen as I would let it go now and run towards the steady state. And indeed, as you see, the system, when I let it go from this scaled down version, it coarsens back to the same size distribution as the one I started off from. And it did so in a very peculiar way. It did so in a way that those cells that have 5 edges, they grow. Those that the 7s grow, the 5s shrink, and the ones that have 6 edges, they stay constant. And that's a very famous law. It's the von Neumann law in foam physics, and this von Neumann law is a fact that's derived from the existence of surface tension and the Gauss-Bonnet theorem. Nothing but that. That's all you need. And of course, the vortex structure that you have in these kind of systems. And that's valid here as well, and you would expect it to be valid if the system is just showing a surface tension-like behavior, and indeed it is. So what's now causing the other type of dynamics? So as you scale your system down, scale your system up this way, it should go back to the equilibrium state by an anti-coarsening process. And the question that we asked, what is that anti-coarsening process that must happen? Here it is. It's kind of cell division, if you like, or inverse topological transitions. That's another way of phrasing it. And that brings it back to the original steady state. So it's the balance between a classical law, the von Neumann law that guides the coarsening behavior, and some kind of instabilities that honestly we don't understand in detail, but they drive the formation of interfaces between the different domains. What's important from all of that for the question of synthetic cell design is what happens with these patterns on a spherical or somehow spherical domain like a vesicle. And what Hendrik finds is that you can tune the patterns on the vesicle to have these polyhedral min-E meshes, and one of them stands really out, right? This polyhedral mesh is just a ring, and that's like a cytokinetic ring in a biological cell. So it's a template now that we can construct by the conceptual insight that those systems show foam-like behavior that could guide the design of these protein reaction networks to actually tune them to form these kind of templates that may act as cytokinetic rings. Themselves, they will not, because they are not able to create forces, but they are templates like the polar cap in CDC42 in yeast cells or other patterns in other cells, on top of which you can then put actin filaments or dynamine or other proteins that do the job. Now, this is another question that I ask. Can these proteins induce curvature? And I think the actual question is not can they induce curvature, but can they generically induce curvature? So by the physics of them binding to the membrane, is there some obvious way by which you should immediately tell, yes, they need to create spontaneous curvature? And we think there is. This is some recent work. And it could explain all the things I'm showing you now. So one phenomenon is the formation of tubulation in vesicles. The other one is, this is really the Min system in a vesicle that studies by Petraschwili together with Reinhard Lipowski, expert in membrane physics. And you see they show these kind of oscillation phenomena. So obviously the Min proteins must deform the membrane interface. And here a very recent experiment by the Schwille lab, where they have on the vesicle also actin filaments. These actin filaments somehow form boundaries, and you see there's blebbing phenomena. The vesicles bleb at locations where the actin filaments are kind of making fences on the vesicle. All right, so that's the phenomena you see, and what could be now a mechanism that drives both an inward type of deformation, so something like endocytosis, if you like, or exocytosis in these kind of vesicle structures. And here's our cartoon. And that's something we're working on right now. So it's Jan Willeke and Hendrik Weyer working on that. So imagine that you have a membrane. That's the membrane surface. That's the lipid bilayer. And now you have these proteins, the Min-D, Min-E, other proteins binding to that surface. But these proteins are not points. They are nanoscopic objects. They are something like 10, 15, maybe 20 nanometers in length. And that means if they bind with a membrane targeting sequence into the lipid bilayer, they will be away from the actual surface by a small amount, by 10, 20 nanometers. And the physics of these proteins should be then formulated not on the surface of the bilayer, but on the surface a little bit, a distance delta away from it. If you formulate it on this distance delta away from it, then it will have the usual free energy that comes from the Helfrich energy with the bending stiffness and the surface tension. That's well-known physics for membranes. And then on top of that, you have a free energy density that will depend on the density of the proteins phi naught, on the surface S naught. And this free energy might just be entropy, right? Rho log rho, or phi log phi. It could also include some kind of Lorry-Huggins terms where you have attraction between the different proteins. And as said, what we need to do now is to actually project this onto the surface of the actual membrane, because there is where the action happens. And as you do so, you will renormalize and change the curvature and induce a spontaneous curvature. And that's very easy to understand because these polymers, these proteins are brushes, right? And if you bend something down, then they have more space. And as they have more space, they can increase their entropy. So there's an entropic effect by which they bend the interface. There is also an effect that it has on the surface tension, because these proteins, they act like a pressure, right? If you have something that you fence and you put in molecules, they want to, in the ideal gas fashion, extend the fences they are confined to. So there's two effects, and those two effects can give rise to different phenomena. If you have surface tension effects, pressure effects from the proteins, if you have something blebbing out, it will favor it because it has more proteins, more space for the proteins to extend, so it has a higher entropy. And that's true whether you do this from the inside or the outside. It doesn't really matter which way you do it, because that's an effect, that's a scalar effect. If, on the other hand, you ask about the effect of spontaneous curvature effect have a direction. It matters whether you bind these molecules here from the inside or from the outside, because in each of those configurations, the polymers want to be further away from each other, so they bend the surface away from it and not towards it. That's not true for the pressure effects. So let me show you simulations Jan did based on that. So that's showing here a two-dimensional cartoon of a vesicle. You have in bluish purple actin, which forms kind of fences. So there's a barrier here that you put on the membrane, and the proteins here are the MinD proteins from our diffusion reaction system. And as I run the system now, you'll see there is an effect. And to point your attention, in equilibrium, if you have the pressure being equal on those two, the pressure will be equal on both sides, right? So you will have nothing deforming these kind of systems. If you, however, have a diffusion reaction systems, you load and unload the interfaces. So you change the number of proteins that you have on the interface, and thereby you will have imbalances of the pressure on the different domains of the vesicle shell. They are fenced off by the actin filaments. And as you see now from the simulation, this loading and unloading of the membrane with proteins creates these pressure effects in the effective Kahnemann-Helfrich free energy functional, and that leads to the deformation of the vesicle just on the basis of protein patterns. So there's a very simple way patterns can create mechanical forces, namely via entropy. That's what you see here. So let me close by the last example, by vesicles on these substrates. And this is just one slide. So here you have a substrate where you have the foam-like pattern that I showed you before. On this foam-like pattern, you have the vesicle here, and the pattern here is on the outside of the vesicle. And that's the configuration on which Petra and her students found the vesicle-like motion, the cell-like motion of these vesicles. Our explanation of that motion at the moment, and this is unfinished work, I would say, is that these protein gradients that you see here, these branches on the surface, they create an adhesion gradient because of the ability of the min proteins to actually attract each other in a sticker-like fashion. Like I mentioned in this recruitment idea, that sticker-like attraction between the proteins gives rise to recruitment. So in a similar fashion, that sticker-like attraction could create a surface gradient. Now, assuming that you have such a gradient in adhesion, you can build a phase field model of your vesicle, including all the essential elements, and you also include the diffusion reaction system, and then on the computer, we can kind of reconstitute the same kind of motion based on the assumption I just said. So to let you go, all the 4 points that I mentioned, I think we are pretty far. We do have this. We have a molecular understanding of those patterns. We know why and how these protein patterns respond to geometry. We have some ideas now why they and how they change these geometries in a generic way. And we begin to understand what kind of different hemomechanical couplings there are between the patterns and between the membrane. And I think it's this arena here that we have to work and focus on to push this even a little further. Let me thank the people who did the work. So Hendrik was the mastermind behind the ideas of having a surface tension in diffusion reaction systems. He also, together with Leshy, worked on the molecular model for the experimental data of Suk-Yun Yun. And Jan Willeke recently studied to implement the ideas on proteins attaching to membranes and giving rise to these entropic effects. And these are our heroes. Without them, we couldn't do what we do. So Petra, Suk-Yun, Nikta, and Kes, they inspire us by their beautiful experiments and challenge us to make theory for real life phenomena. Thanks for your attention.
Thank you very much.